morning, viewer. Welcome to our Globe TV is amazing news for our viewer. Yeah, DNA aptamers are powerful molecular tools in biosensing and bioimaging, also therapeutics. However, a limited understanding of their tertiary structures and binding mechanisms hinders their further optimizations and applications too. Adenosine triphosphate, in the short form ATP, a central metabolite in cellular energy metabolism, is a key target for aptamer development. So, a DNA aptamer 131B has recently been reported to bind to one molecule of ATP with a dissociation constant K5 of 2 to the power 5 nanometer. However, the structural basis for ATP recognitions by 1301B remains unclear, lacking guiding principles for rational optimization. In a study published in PNAS, a team led by Professor Tan Weihong, Professor Han Da, and Professor Guo Pai from the Hangzhou Institute of Medicines of the Chinese Academy of Sciences determined the tertiary structures of a DNA aptamer ATP 1:1 binding complex, revealed the recognition mechanism, and engineered and optimized DNA aptamer with a sub-micromolar K5 for ATP binding, which exhibited the highest affinity reported for ATP binding DNA aptamers to date. Researchers determined the solutions to nuclear magnetic resonance structures of a certain variant 1301B V1 in complex with ATP. The structure showed an I-shaped architecture where ATP intercalates into the binding pocket formed by two internal loops as stabilized by hydrogen bonding with guanine and stacking interactions with neighboring bases. In addition, researchers showed that Mg2 plus ions facilitate 1301B/V1 to form a semi-folded structure and further stabilize the binding complex by neutralizing the negatively charged phosphate groups of DNA at ATP2, which demonstrated that the aptamer recognizes ATP through an adaptive recognition mechanism. Behind this exclusive report, Globe TV, Delhi, Bharat.